We have 11 and 13 participants, and therefore a big number online, still online with us. Thank you so much. Welcome back. And a few words on Cedric Notredame. Cedric Notredame got a PhD in bioinformatics in 1997 at the MBL MBI, and so he was out of Heidelberg. And then he went through Europe, first with the postdoctoral position in Lausanne, and then as an assistant professor in Marseille, and then as a CNRS investigator, and as an assistant professor at the Lausanne University. And there he was also at the Swiss Bioinformatics Institute as a group leader. Finally, in 2007, he became a senior group leader in the Bioinformatics and Genomics program in Barcelona at the CRG. And then, since then, he frequently came to Bologna to give us courses. So he's a professor practically of the Master in Bioinformatics as well. Now, obviously, the name of Cedric Notredame is first of all linked to the tea coffee tool that everybody has been using in life, including us, for several reasons. And he has now a group, and the ongoing projects in the group include improvement of RNA multiple sequence alignments, a very difficult task, post-processing of multiple sequence alignments, benchmarking and validation of multiple sequence alignment methods, combination of sequence and structural information, protein structure comparison, etc., etc. So many tools that are freely available to the whole community. For this reason, is fairly well known in the international societies and of all the researchers in bioinformatics. And as I said, we are proud to have Cedric Notredame in our staff here at the Bologna Master in Bioinformatics. So welcome back, Cedric. Also, thanks for having our students in your PhD programs. And the mic is yours.
Well, thank you so much, Rita. And I'm so happy to be back, and I'm so unhappy not to be back. One of the good things in my life is to visit Bologna every year. And actually, if some of you read some of my blogs, I was joking, but this is actually true. There used to be a time, and it is now coming back, where every year I will travel to Bologna to teach in the master with my wife, actually, who joins me for some reason. She always comes when we go to Bologna. And then we will go back to Spain with a few more kilograms and one PhD student. And so far I have you have already given me 3 PhD students, Rita, and all of these have been amazingly successful, including one of them, Evan Flodan, who very, very successfully launched his own company in Barcelona on stuff he did in the lab. And these guys have just raised 5 million euros with 2 Chan Zuckerberg grants. So that's something quite noticeable. They did a company on Nextflow, which I will talk about. So yes, we go back a long time. I think it's been more than 10 years. I think the first course in Bologna was probably almost, that doesn't make, well, you, Rita, you don't age, but normal people like me, yeah, that doesn't make anyone younger. Anyway.
I close the video, okay? So
and
fantastic.
I will share my screen. And I will share my screen. And so, can you guys see my screen? All right?
Yes.
Yes? Okay, I'll just try to go in full screen. You should see it in full screen. Is it fine?
Yes, perfect.
Yes? Okay, excellent. So, the title is a little bit provocative here because I'm talking about multiple sequence alignment in the post-sequence era. And so, there used to be all this thing in the post-genomic era, you know, when the first genome had been done, you had to resequence the genome. And so by the end of the 90s, beginning of the century, there were all these talks about post-sequencing era, blah, blah, blah. And I'm arguing here that we may be actually entering the post-sequence era, an era where rather than using the sequences to align them we'll be probably using the structures to align the sequences and then doing any kind of analysis we need to do in the sequences. And that major shift is actually something that has been made possible by AlphaFold 2 or AlphaFold the second, because it's really the emperor of our life these days. It's a very interesting and important transition, and I know that throughout all of this series of lectures, you're gonna look at all the aspects and all the angle through which AlphaFold can be looked at. And as you know, Rita here was one of the pioneers of secondary structure predictions using all of these artificial intelligence methods back in the 90s. And things have not changed so much, actually. Conceptually speaking, pretty much everything. Well, you're going to have Burkhard Rost also, who was a pioneer on these things. And you will see that things have not changed so much. What has changed is computer power and data availability. It's really what is making a big splash. But this is happening now. All of these things are crystallizing at the same time. And I think we are extremely privileged to be living at this special time with all this conjunction of things. Now, this thing is a course, and so I apologize to those of you who heard my course last year. A lot of it overlapped with what I'm going to say this year, with a few add-on here and there. And so we'll go back to some very basic things. And one of the most basic concepts that I like to convey here is that there is actually right now, as we speak, a race going on. And this race is between data and computer power. And it's a real race because we cannot let data grow faster than computer power. Otherwise, we will simply not be able to afford the amount of money we need to analyze our data. And that's a real issue. It's an issue that is becoming more and more pressing because of all this sequencing, of all the improvement in sequencing. And what you have here, what you can see here is the growth. So this is all exponential, okay? So all of these things, what is even worrying is that sequencing is not a straight line here. Sequencing is an exponential, which means that when we look at this graph, we can see that every once in a while, and at least during this period, sequencing was super exponentially growing, meaning that the amount of time it will take to double was shrinking a little bit every day, okay? And when this amount of time it takes to double reaches zero, then something happens. It's a new Big Bang, or I don't know, but something has to happen. And what is important is that even if we extrapolate this to a straight line, which it should be, the two lines don't have the same slope, which means that one is doubling faster than the other. And that's what I'm calling a race. And it's not a race we are currently winning. It's actually a race we, data scientists, are losing. Now, things got much, much worse actually since 2011, and they got much worse because you see the curve here. All of these new generations of sequencing, all of the next generation of sequencing, always make things a little bit worse. And it is fair to say that our appetite for sequencing is impossible to satisfy, because eventually we'd like to be able to sequence every cell in our body. And even more than this, we'd like to be able to sequence the transcriptome of every cell in our body every minute. And in one of the journals where I'm editor, I've just received this interesting paper about something that is ongoing, in vivo sequencing, which is something that will probably become possible in the years to come. So that's even more data. These are incomprehensible amounts of data that are going to be provided to our appetite. So what? It's really the match of the century, right? On one side, you have Moore. You probably all know that Moore was an engineer, I think in Texas Instrument, I think he was, or no, Hewlett Packard, I think. And he figured out at some point that every 12, 13 months, the power of CPU was mechanically doubling. Nobody knew exactly why. And the Moore law has been given for dead quite many times over these last decades. But, every time, because of some new innovation, the capacity of engraving things a little bit thinner, the capacity of nanotechnologies. Yeah, we keep follow the law of more. And this gentleman here is Sanger, is Fred Sanger. Fred Sanger, if you look up his medline, Fred Sanger has something in the order of 25 research papers, no more than this. Out of these 25 papers, he managed to produce 2 Nobel Prizes, which is an unusual average. So that was really, that's the epitome of a highly talented scientist with no hype, just pure stuff. And so here we go. CPU power is doubling every 18 months. Gene sequencing capacity is doubling every 7 months, which means that all things being equal, if you want to keep being able to analyze your data every year, data that is produced by your sequencers every year, if you change your sequencers every year, and if you change your computers every year, well, you're going to have to spend twice more, roughly, a little bit more, twice more on computers every year to be able to keep up with your data. And if you remember the story of this humble man who asked the king of Persia to give him only 1 grain of rice on the first case of the checker, and 2 for the second, and 4, and so on and so on. And the Shah of Iran said, "Oh yeah, of course," and then realized very rapidly that all the rice in the universe will not be enough. Well, if you keep having to double the amount of money you spend in your computers every year, you are going to have problems with the head of your university very, very soon. So what? There are 2 solutions, really. The one that I was mentioning, buying twice more computers every year, and the second solution, which is the only sustainable solution, is to have efficient algorithmic scale-up, making better algorithm. There is no alternative, I think. And this being said, some of you may say, okay, but we have all this data. Is it going to be of any use? Maybe we could just forget we have the data, and maybe nothing will happen because maybe the data is not useful, really. And fair enough, but I will say that there is something extraordinary with data. Data fuels biology because biology is based on comparisons. There is nothing in biology that does not become soluble at some point in sufficient comparisons. And in fact, if you think about it from a conceptual point of view, comparisons are the only way of revealing processes that are unknown to us. We are like these people trapped in Plato's cave. We are chained, and there are the puppet masters on the other side. And in our case, we don't even see the puppet masters of the cave. What we see is the product of a transformation. So what we're interested in is the process that created this transformation, but we only see its product. And our only way to reconstruct the machine that has been carrying out this transformation is to compare many alternative products of the transformation. That's what we have access to. And this is exactly what Darwin did. Darwin looked at all these birds, he compared the shape of their beaks, and came to the conclusion that there had to be some unified process that could be described by its capacity of generating the type of diversity Darwin had observed. Okay? And as far as I know, I cannot think of a better and more powerful data structure to reveal this invisible process than a multiple sequence alignment. Now, this is a toy example I've been showing for decades now, and these are just a bunch of sequences that have been aligned. They have been aligned so as to put in the same column residues that are believed to be homologous to one another. Or, put it another way around, when the alignment is correct, all the residues in the same columns will be homologous. Homology is a term coined by Darwin that relates to characters having a common ancestry, characters whose similarity can be explained by a common ancestry. Okay, and so that's why actually we often call these things models. Mathematicians are sometimes a little bit confused when they hear us calling multiple sequence alignment models. Why are these things models? Well, for the very reason that they are actually predictive tools. When I am making such an alignment, I am predicting something in the past, actually. There is this joke about predictions are very hard, especially about the future. But as it happens, it's often just as difficult to predict the past as it is to predict the future. I think it's Bohr who said this. Yeah, predictions are difficult, especially for the future, those about the future. But they are difficult also about the past. When I'm making a column here, I'm actually predicting that these prolines here correspond to the same common ancestor that occurred in the past. Now, this is a very, very powerful way of looking, of integrating your data. And in fact, it's one of the most powerful way of integrating huge amounts of data and making sense, ordering this data, giving names to your column, calling this an active site, and seeing it shared across all of the other members of the family. And this is, I'll just go back. This is very, very well reflected by the fact that you probably have seen this paper in Nature. I think it was in it's 2014, a few years ago. And so they said, they made out this thing. If you were printing every page of every published paper since, middle of the 19th century, something like this will give a pile of paper, the first page only, a pile of paper about as high as a Kilimanjaro. And so that's how we figure out that the Kilimanjaro is about 5,200 meters, something like this. And along this pile of paper, the top 100 papers are here. It's very, very tiny. And among the top 100 paper, you probably have heard about protein staining, which is the most cited paper ever. And close enough, at number 10, we have the Cluster W paper. Now, Cluster W is a multiple sequence aligner that was developed at EMBL in the 90s. Actually, this was developed by my PhD supervisor, by Des Higgins. And if I had been in the lab maybe a year before, I will probably have been able to contribute a little something to this paper. I hope I will have been on the paper as well. But I'm fine. I'm not here. I'm somewhere on this file here. But that's okay. But it's great. What is very interesting here, of course, is I'm biased on this. I love this paper, this Nature paper, because it says it's all about methodologies. Methodologies change things. And my colleagues who work on biology and try to establish complex biology mechanisms, no, these citations are overrated. And of course, we are both right. If you are a biologist and you unlock a very complex mechanism that makes it possible to understand a lot of things, you won't be very much cited. You will be cited by your immediate neighbor, but you will make a lot of things possible. So for biologists, pure biologists, citations are not necessarily a great.
Cedric, you are muted.
I've been muted. I just had to show my. Should I?
Okay, now it's okay.
When did it stop?
Few sentences ago, not very much ago. So.
Okay. I'll just. What was I saying again? I was talking about. Sorry, I lost my thread suddenly. Anyway, it doesn't matter. So yeah, I was talking about citations that matters and don't matter. And so if you're a biologist, if you look at the Sanger, at the Watson and Crick paper, I think this paper has about a few thousand citations, which looks silly because, this was the most important discovery ever. But the other thing, it was such a matter of fact discovery that it stopped being cited very fast. BLAST has much more citations because still today, 20 years after, if you use BLAST, you cite it, and you cite it because it's a very technical tool and everybody is not supposed to know about BLAST, okay? So this is just to put a grain of salt on these things. But all things considered, these things are very, very important tools and they are very highly cited for these reasons. And as I was saying before, if you do like me and you develop tools, you want to be cited, because if you develop tools and you have no citations, it means that your work is actually not as useful as it should be. Oops. I'm just trying to Yeah. And so these are just some of the many applications of multiple sequence alignments. And we're going to see in a few minutes that AlphaFold is one of them. But basically, whatever you want to predict from sequences, you have to use a multiple sequence alignment at some point. This one here, Polyphen, is interesting because Polyphen and SIFT, these are the most medical application of multiple sequence alignment. Situations in which it is from the evolutionary constraints that you can read in your multiple sequence alignment that you're going to figure out if a mutation is actually a dangerous mutation or not. Okay, and of course, phylogeny, which is something I'm extremely interested in. So what do multiple sequence alignment tell us about biology? That's one of the things we have to try to figure out. And I apologize for those of you who already know this story, because I've been telling it for so long that probably most people have heard it, but people who know me have heard it right now. But this is actually a true story. It's a story. It's a Second World War story. So Abraham Wald was an engineer during the Second World War, and he was asked by the army to help increase the survivability of military planes. You know, the militaries have this gruesome statistic that if you lose 1% of your plane every week, then you have to produce so many new planes and train so many new pilots and all these kind of things. And of course, you cannot overprotect the plane, otherwise it will be too heavy to take off. So what can you do? Well, what Walt did was very smart. He drew a cartoon. So this is a modern plane. That's exactly the same idea. He drew a plane cartoon, and every time the squadron was back from missions, he will report the location of the bullet impact. So this would have been Jim coming from Thursday mission, and this would have been Peter in a different aircraft coming back on the same mission, and on and on and on. And after some time, the bullet impacts accumulate. Now, when I give this as a lecture, I usually make a pause, a dramatic pause, and I ask to the students, and so what will you do with this information? And almost all the time, I have a student brave enough to give the wrong answer, which is the one most people give actually. And the obvious wrong answer is, well, you will protect every piece that has been hit by a lot of bullets. And of course, this is a wrong answer. Why is this a wrong answer? Because why is it that you can see this bullet impact here? Well, you have two necessary conditions. The first one, obvious, is that the plane had to be hit by a bullet on this location. But the second one is that the plane also had to come back, otherwise you will never have seen the bullet impact. Now, when I see my plane, when I have the wing here with so few bullet impacts, it's hard to tell. I don't know if there are few impacts because the way the plane presents itself and all these kind of things mean that the wing is a little bit less exposed, or because indeed, whenever the wing was hit, the plane went down. But when I look at this suspiciously depleted spot here, it's more interesting because here I have a background, and the background is telling me, oh, yes, you don't want to be sitting on the back of the plane. But overall, as far as the plane is concerned, it doesn't seem to matter except here. And that's the kind of information you'd like to use. The kind of information about this part here is very exposed. This is what the background is telling me, and it has to be protected. Now, what we have in this little scheme is biology on a chart. You know, we have a background, we have a signal, we have everything we need. And that's exactly what a multiple sequence alignment is. Every line here is a sequence, and every sequence is actually a plane. And what we have here are the variation, the mutation, and each mutation can be seen as a bullet impact. For instance, on this location here, on this column, let me look for this column here, every residue is different. Leucine, methionine, serine, valine. So obviously, just like in the scheme of planes I was showing before, this is a position that can tolerate bullet impact, that can tolerate mutations. While on the other hand, this proline here is very, very highly conserved, which means that probably you should not touch this proline. Probably every time this proline was touched, something terrible happened to the protein and everybody died. This is the equivalent of touching the engine in your plane. This is the equivalent of this region that is depleted of mutation in your plane. It's a region. It's not that this region does not receive mutation. It is that when it receives mutations, something happens. Now, it may come as a surprise that protein can evolve under this kind of constraints, whereas in yeast, for instance, you can remove most proteins and the organism will manage to survive, maybe not so well, but it will manage to survive. So it's interesting, and that's something that was shown by one of our colleagues a few years ago that had been suspected for a long time. But this was shown very nicely by Ben Lenner, that actually the proteins are not only important by their function, they are also important by their innocuity. You know, it's not in a cell, it's not that you should be useful. That's good. That will protect you a little bit. But it's much more important to be harmless than to be useful, because you can be useless, but you cannot be harmful to the cell. You cannot be a toxic poison to the cell in which you're expressed as a gene. And most of the conservation, most of the constraints we have here, they're not so much about maintaining the function. Many of them are, especially in the active sites, but they are very much about maintaining the fold. Because if as a protein, you do not fold correctly, then you precipitate. And most of the time, if you start precipitating, you're going to kill the cell. And so that's going to be a lethal phenotype. And so it's an irony that proteins that are not necessarily essential to the cells nonetheless evolve under a strong pressure, in the sense that they have to be useful, otherwise they will rapidly be lost, but they may not be extremely important. There may be something where the protein, the cell phenotype will decrease a little bit if it loses them, but if they start misfolding, the cell is going to lose it entirely. Now, I'm showing here AlphaFold, and unfortunately, I won't be talking too much about AlphaFold, but what you have here is the role of MSA in AlphaFold. So you will hear about AlphaFold by many people who understand it much better than I do and ever will. But I will give you my take on AlphaFold, and I will tell you how I see it and how I consider it. You've heard and you've seen in all the newspaper that computer scientists have solved the problem of protein folding and all these kind of things. You know, this is not true. We have not solved the protein folding problem. Nature has solved the protein folding problem. Nature knows how to select protein that fold correctly and get rid of those that do not fold correctly. As I was showing before, nature has this capacity. What AlphaFold has done for you is that it has collected all the experiments made by nature. And in fact, the multiple alignment we were seeing before, it's a collection of experiments made for you by nature. You have the results of thousands, billions of experiments. And so AlphaFold is using this multiple sequence alignment, and it is combining them with other information, structural information, okay? And this combination, we had known for a long time that this combination was a secret of understanding protein folding, but we didn't know how to do it. Or if we knew how to do it, we never managed to do it. And these guys manage. And so I usually describe, or at least the way I describe AlphaFold to myself and neural networks in general, is that it's liquefier. It liquefies data in such a way that bulky data, complex data that had to be handled in specific way, suddenly everything becomes manageable in a uniform way. You know, you've liquefied your experimental structural information. Here it is, ready for the cocktail. You've liquefied the multiple sequence alignment data. Here it is, ready for the cocktail. You can mix these things, and here is what you're going to get. You're going to get these amazing models. Now, I'm very sorry that my multiple sequence alignments are not those used by AlphaFold, but I have hope that some of the things we are doing could hopefully lead to even more accurate models. But I'm extremely proud that one of the guys on this paper, Martin Steiniger, who is actually the guy who provided the multiple sequence alignment methods, was actually staying in my lab as a visitor when he started working on these methods shortly before he went to Korea. And so it's one of the things that shows some kind of continuity between all of these things we are doing. Actually, Martin developed very efficient database searching procedure that can be seen as stacking multiple alignment. And these are some of the things used here when he was working with Jonis Sudding. And that was the time he visited my lab at the end of his PhD. He was a long-term visitor. And so what I'm showing you here is that in order to make these very super accurate structural predictions, well, you need to tap into evolution. You need to have two types of experimental data. You need to have the crystal data, and you need to have the natural experiments. You could play games and do all the possible mutations, and people are doing this quite a lot these days. You know, you've heard about this exploration of mutational landscape. But, you know, with a multiple sequence alignment, you have it all done for you by nature. And it's just costing you the sequencing. So is it possible to do useful biology with very large multiple sequence alignments? I hope you will agree with me that AlphaFold 2 is useful biology. Actually, one of the things with AlphaFold 2, and one of the things that is driving everybody crazy, is that aside from Martin, I do not know any of these people as biologists or as genome biologist people. They're all computer scientists. So it's something quite remarkable that all of this crowd of computer scientists just came together and using all this brute force computation with the state-of-the-art methods managed to crack this problem in such an elegant and efficient way. Okay, so it's something remarkable. This will, you know, I'm one of many saying this, but this will really be one of the landmarks of modern biology. Now, back to the question, is it possible to do useful biology with large multiple sequence alignments? Well, if you read some of the stuff I wrote, especially in Bioinformatics for Dummies, which we wrote about 15 years ago, I said, no, they're useless. You don't need big alignments. But at the CRG, I changed my mind. And I changed my mind because of this guy, because of Fyodor Kondrachov. And so Fyodor is an evolutionary biologist who is interested in epistatic interaction. He's interested in protein evolution. He wants to know if proteins could evolve to the entire protein space, if they have some kind of constraints preventing them from exploring this entire space. And Fyodor managed to convince me. So, well, it did not convince me. Fyodor said, I need very, very big alignments because there are very subtle effects that I will only be able to measure on huge alignments. You know, you can think of an alignment as a kind of a time machine, or even better, as a telescope. You know, when Hubble and the new telescope that just launched, I can't remember its name, when these telescopes look deep into the space, they're actually looking deep into our past, because as we all know, the light of the very distant stars is only reaching us long time after it was emitted, years, centuries, millions of years after it was emitted. And in a way, the multiple sequence alignment, because it reflects events that occurred in the common ancestor of all these proteins, it can be described as a time machine as well. It can be described as a data structure that is reconstructing the past for us, for our benefit. And from that point of view, if you want to look very, very deep, you need a very, very large multiple sequence alignment. And if you want to figure out very, very subtle evolutionary patterns that are happening very slowly, but very steadily, you are going to have to look at very large multiple sequence alignments. And this is exactly what Fyodor did in this paper with our help for the production of these very large multiple sequence alignments. Now, if you look at the paper, we were not, the method we used for these very large multiple alignments was very ad hoc. And it turned out that these were relatively easy alignments, because these were alignments that could have been done by hand. And Fyodor actually initially tried to do them by hand. It's just that you had a few gaps here and there, and you had a few things that needed to be done. And this was complicated. So Fyodor asked us to do the alignments. And as it turned out, all the aligners were producing terrible results, even though the sequences were nearly identical. They were highly conserved orthologs from mitochondria. Most of them were highly conserved ortholog from mitochondria. These things evolved slowly, but even though with 1,000 you had a problem to do it automatically, and that's what got us into thinking about scaling up these things. And I'll be talking about the follow-up of this project that we did with Theodore as well a few years later. Now, having a big multiple sequence alignment, that's the thing. Now we can align up to 1.5 million proteins. This is the largest alignment that we have been able to assemble a few years ago. This is the ABC transporters in PFAM. It's smaller now because they had to break it into subfamilies. But at the peak of its height, it was at 1.5 million sequences. And not many methods are able to align this. And that's one of the things I'll be showing you later. So as I was telling you before, these very, very big alignments are great because they allow you to really do precise residue interaction or protein-protein interaction analysis, because you see coevolution in action. You can use these things to do smart things, and that's one of the examples that is shown here. It's Desigin to do that stuff, and he actually evaluated the quality of its alignment, of his alignments, by measuring how good was the structural predictions based on these alignments. Okay, because good multiple sequence alignments allow the prediction of 3D contacts. We knew this before AlphaFold 2, but AlphaFold 2 confirmed it in a spectacular way. Okay, so we have more and more and more and more data. And I'm sure you have seen this image this morning with Roderick Guigo, who was this morning's speaker. Yes, you have. And so it's huge because we're talking about 1.5 million species to be sequenced. And, you know, one of the things that strikes me here is that for years, I've been calling myself an evolutionary biologist. And I've been reading about evolutionary biology. I've been studying about evolutionary biology. But as it turns out, evolutionary biology has not really started, you know. Evolutionary biology started with Darwin, and Darwin made all these nice phenotypic analyses, comparison. I understood the nature of the underlying mechanism, but Darwin had no clue this was mapped onto DNA. It took almost 50 years to really nail it as being the substrate of this evolution being DNA. And it took 50 more years to really figure out how DNA was working and what was the actual quintessential nature of DNA. But we didn't have DNA, you know. We only had the first genome in 98, and this was a tiny genome. This was an E. coli genome, and then we had the first eukaryotic genome a little bit later, and then the human genome was kind of, the human genome was just ready last year, actually, because reasonable chunks of the genome were hard to sequence, the euchromatin and these things. But anyway, well, on paper, we had the genome at the beginning of the century. But you don't do evolutionary biology with one genome. You need a lot of genomes to do evolutionary biology. And so it is just now with this project, and when this project will have delivered, that we will be able to do evolutionary biology. And if you know your epistemology, if you know your history of science, you know that science works like, especially experimental disciplines like biology. You have observations and you have a theory that accounts for your observations, and you're happy. And then suddenly you change the apparatus with which you do your measurements. You know, you go from go from looking at the stars with your naked eyes to looking at the stars with telescopes. And this allows you to make much more precise, newer observations. And you realize suddenly that the theory you had, well, doesn't fit all the data. You know, plenty of your observations do not agree with your theory. And that's where you know you need a new theoretical framework. And I'm no big genius by saying that once we will have the 1.5 million species that are contemplated here, biology as we understand it today will have to be redefined in something that I guess, will not be that different from Darwin's initial insight, but it will be extremely different when it comes to the fine details. And again, just like with AlphaFold, we live at this very moment of biology. We live at a moment where this kind of amazing data is being acquired. So that is something extremely important and extremely, I'm just keeping an eye on the time. It's extremely important and extremely exciting to be doing this kind of things. So now let's talk numbers. If you have 1.5 million animals, and if you look at your multi-member, protein families like kinases, for instance, you're gonna have about 200 to 600 families in your average eukaryotic genome. Well, if you have 1.5 million sequences, you're looking at something in the order of 1 billion sequences. That's huge. And we have no clue on how we will be able to deal with this. Now you may argue, but what are you going to do with these very big alignments? What are they going to contain? I don't know. I only know one thing. I know that we are going to have to classify this data. And the only way I know of effectively classifying this data is to make multiple sequence alignment, is to declare equivalencies between all the residues. If you have these 1 billion kinases aligned together, you will have vectors, you will have amino acid vectors that will contain 1 billion amino acids. You're looking at the beginning of life on planet Earth, and you're looking at an incredibly complex collection of scenarios by doing this. And it is the notion of equivalence, it is a notion of homology that is driving all this. Without homology, you have no organization of this data. And once organized, this data is a kind of map. It's a map that you can use to look at living things. So it's important and it will be useful. How good are our multiple sequence alignment methods when scaling up? Now that's an interesting question. And that's probably us going a little bit closer to what we have to deal with today. Before we do this, I'll do a brief introduction of how multiple sequence alignment methods work. What are the little engines behind? And you will be surprised to see that it's very simple, actually. So in an ideal world, we know how to align two sequences. We know how to, if you have something like a substitution matrix, a palm or a blossom, you have a gap penalty. Well, we have something called dynamic programming. It's an algorithm designed by a computer scientist in the 50s. Bellman actually was the inventor of the word, coined the word dynamic programming. And then much later, it's actually a Dutch scientist called Dijkstra. So if there is Dutch here, you can unmute yourself and pronounce this correctly. It's this guttural Dijkstra or something like this. And I encourage you to read the account of his discovery. So that's a source to think algorithm. So that's an algorithm, this dynamic programming we use all the time. It is the algorithm you use on a GPS. When you go from point A to point B, this is something that is going to figure out the shortest path, okay? It's not a complicated algorithm, it's a quadratic algorithm. So if you have two sequences, it's going to take an amount of time that is proportional to the product of the length of your sequences. And I was talking about Dijkstra because in the Wikipedia, there is an interview of him, and he says that it's an algorithm he invented without a pen and a paper. He was walking in the street and he had this intuition, and he said, and it is simple because if you do algorithms without pen and paper, they have to be simple, okay? I think it's a nice story. I don't think it's any way simple, but I think Dijkstra really was a genius. And anyway, it looks, on paper, you have this algorithm to align two sequences. Well, why not aligning three sequences? And yeah, okay, it is not anymore quadratic. Now it's cubic. And rather than having to figure out the optimal alignment here, you figure it out in 3 dimensions. Unfortunately, it's not that simple. If you go cubic, you need, if your sequences are 1,000 residue long, you need more data. And actually, we can play the game. This is how we dress students in our lab so that we recognize them. And you can see here a student who has just aligned 2 globins, and it took 1 minute. So the student is surprised because, you come here for an internship and it only takes 1 minute. Well, what is this? So I was an ambitious student. He went for the 3 globins problem. And with these algorithms we had before, for 3 globins, you need 2 hours. And for 4 globins, it's 10 days. And 10 days gazing at the computer, it's an old story. So in these days, the screens were not very good. So you get sort of eyesore by looking at the Hertzian screens. But, you know, the guy was ambitious, went for a PhD, the 5 globins problem that took 3 years. I told that he has decided to go for the 6 globin problem for his postdoc, which is going to take 300 years. And my guess is that if this person goes for a 7 globin problem, well, that's 30,000 years. And for the 8 globins problem, we are at 3 million years. And I have no idea. I mean, at the current pace of things, I need to be convinced that a carbon-based life will still be a thing on this planet. Anyway, I'm not 3 million years, and I've aligned thousands and thousands of sequences. So I have to have a trick. And that trick is about the progressive algorithm. The progressive algorithm, so an exact method will be too slow, and we will therefore use a heuristic algorithm. And out of this heuristic algorithm, the progressive alignment is the most popular. And so the most popular implementation is a cluster W algorithm I mentioned before. It's not strictly speaking a progressive algorithm, but it's close enough. And so it's even worse than being a heuristic algorithm. It's something called a greedy heuristic. In French, these greedy heuristic are called gluttonous algorithm. And that probably rings a bell in Italian. I guess glutton probably means the same thing. And so these are algorithms where I usually give this explanation to my students. I say, you know, imagine you go to the cafeteria, you're very hungry. Oh gosh, you're hungry like you've never been hungry. There are no forks and no plates. What are you going to do? You got to eat with your hands as you go across. And so you move to the left and you see potatoes. And you know, potatoes are fine. And you're so hungry, you stuff yourself with potatoes. And then you move a little bit more to the right. And it's caviar and lobster and all the things you love so much. But you're not hungry anymore because you stuffed yourself with potatoes so much a minute ago that you have no room left for the lobster and the caviar. And that's a greedy algorithm. And that's exactly what greedy algorithms do. They take the best decision at a given moment without making any provision for a better decision that could have been taken waiting for new data. Okay? And so the progressive algorithm is by essence a greedy algorithm. And we're going to see why in a second. But it's fast, super fast. So at the beginning of this algorithm, like any time you'll be doing sequence alignment, you have clustering. And that's actually a lot of work is still ongoing on the clustering side. And in fact, I would argue that most of modern data science is clustering. In a way, even deep learning can somehow be described as a labeling exercise and therefore a clustering exercise. So that's how important clustering is. You know, these machines, these self-driving cars, it's all about segmentation of the images to distinguish the road from the rest. And that's also a clustering algorithm, you know, putting together everything, you know, drawing boundaries between points. So clustering is really the most essential, the most central activity in computer science, in data science, and probably in our lives. Probably we spend most of our time doing some kind of clustering. And so in the original paper by Paula Hogeweg, and it's a nice story because for many years, everybody was citing different scientists for this paper who reinvented the algorithm a few years later. But to show that it is not true, that bioinformatics has no memory, slowly but surely, everybody has started citing as the original inventor of this method, Paula Hogeweg, a Dutch scientist, actually, another Dutch, whom I understand is still active. I think she I don't know her personally, but I think she's still active. I don't think she's retired yet. Anyway, the bottom line is that she figured out that you can cluster your sequence using a regular clustering algorithm, UPGMA. Now people prefer using neighbor joining. And once you have your cluster, you can do your alignment. And how are you going to do this? Very simply, you're going to align things 2 by 2. And so the first thing you'll be doing is align, it's moved a little bit, this was meant to be here, but it doesn't matter. the first thing you're going to do is align the blue sequences here because, you know, they are blue. No, because the clustering is telling you that they're a bit more similar. And so you'd rather align the blue sequences. Now, one thing that is very important to realize is that pairwise alignments are not perfect. If they were perfect, there will be no multiple alignment problem. You just align all your pairs of sequences and you will plug all of these pairwise alignments together. That's not the way it works. The way it works is that if your two sequences are 100% identical, their alignment is trivial. If they are 50% identical, you're going to get a pretty good alignment. If they are 20% identical, your alignment will most likely be a random alignment. You have no way to tell if it will be correct or not, okay? And that's an issue because the optimal alignment of your two sequences exists. You know, if you have two sequences that have 20% identity, and you give them to dynamic programming, you're going to produce an optimal alignment. It is just that this optimal alignment will be very different from the correct alignment of these two sequences. And the correct of the alignment of these two sequences will be the one in the multiple alignment, which means that your multiple alignment is actually a collection of most likely suboptimal pairwise alignments. And that's what makes it that's one of the reasons it's actually an NP-complete problem. I don't think I mentioned this before, but the reason we have to use heuristics is because this problem is NP-complete, meaning it is non-polynomial. It's not NP-complete, it's NP-hard. It's the correct term in the computer science. Anyway, you have here, so you're going to align the blue sequences with your dynamic programming, 1 minute. Then you're going to align the red sequences with your dynamic programming, 1 more minute. And now you have these two little alignments. And you can treat, so conceptually, imagine you were turning this alignment into a consensus sequence. Well, then you could align the two consensus sequences you have here in 1 minute. And so rather than spending, I think I said, 4 sequences was something like 10 days. Rather than spending 10 days to align your 4 sequences, you are going to spend 4 minutes aligning your 4 sequences. 3 minutes, sorry. Of course, you know, I feel like in Barcelona, we have this nice avenue called the Rambla, where you can buy Rolex watch for 10 euros. But, you know, it's not really a Rolex, you know, it has to be something different. And here, when I'm telling you that I can do in 3 minutes something that will take 10 days if I were doing the computation right, you know, that's somewhere I have to be cutting corners. It's not possible. And of course, I'm cutting corners. And where am I cutting corners? Well, because when I'm aligning these guys here, I'm not using information in the blue sequences. And if these two sequences are less than 20% identical, or if some regions here are difficult to align, contain ambiguities, well, I don't have access to the blue sequences to deal with these ambiguities. I'm stuck with these ambiguities. And that's precisely the reason why this algorithm is greedy. Here, we are taking decisions, you know, like eating potatoes. Let's say this is a caviar, and these are the potatoes, and, you know, we're eating the potatoes here, and the caviar will come later. And if I have to change this alignment with information, with the caviar information, well, it's too late. And that's why this thing is actually greedy. Okay, but it's very effective. It works very, very well. And the thing I've just shown you is just a canvas. There are many, many, many variations. There are hundreds of variations, actually. In fact, with a student of mine, with Maria Chetsu, we wrote this nice review a few years ago where we played, these are the aligners, and these are, you cannot read very well, but these are all the various algorithmic ingredients. And you can see here the guide tree. So all of these alignments except one here, which is like La Jolla, but all of these things are progressive algorithms. They use a guide tree, they use a pre-clustering, and they use a clustering to guide the order in which the sequences are being aligned. Okay. How accurate are these things? Well, we need to define accuracy. You know, it's not like there is an absolute accuracy. There is an objective function. There is something we try to optimize, and typically we try to optimize similarity. We try to produce the alignment that will have the highest level of similarity. That's mathematical optimality. And this thing is called an objective function. And unfortunately, because we do not really understand, even though we can compute good structures, we do not really understand the relationship between sequence and structure. We cannot turn it into an equation, into a closed form equation, which means that, and that's an issue we have here, which means that we need a way to assess the biological quality of an alignment using external information. And in this case, we are going to use structure-based reference alignments. Why are we going to use structure-based reference alignments? Because structures look as if they were evolving slower than sequences. It doesn't mean they are. You know, everybody is in the same boat and everybody is rushing toward the end of times. But structures are much more resilient, meaning that as time goes by, folds tend to be conserved, which is actually one of the things AlphaFold is using. And so it's an irony that if you take two structures and you start to try to superpose them, the superposition is, under most of its formulation, it's an NP-hard problem. But the signal contained in the structures is so strong that most of the time you will get a distant superposition from which you can extrapolate a very good sequence alignment. And these structure-based sequence alignments are currently considered the standard of truth. Okay, and so what do I have here? I have two residues, number 5 along this line and number 7, and that's my target alignment. These guys are aligned together, and my structure-based alignment can be expressed as a collection of pairs. You know, an alignment is actually a collection. It's a sum of all the pairs it contains. It's just that all of these pairs are compatible into one final alignment. And so the structure-based reference alignment here, that pair here will be considered correct because you find it in structure-based reference alignment, while this pair here will be considered incorrect because you don't find it. You don't have a line residue number 9 with residue number 8 here, and that pair here does not occur in my reference. So that's a no. And therefore, I can express the accuracy of this target alignment as the fraction of pairs it contains that also occur in the reference alignment. And this thing is called the sum of pairs. That's how we determine how accurate a multiple sequence alignment is. So using these kind of things, people have been measuring the accuracy of alignments. Of course, for this, you need the reference data set. This is some stuff we did for a review a long time ago. I find that BB11 of Balibase, it's a reference database. It's pretty good. And so here you have almost 30 years of research stacked. And so these guys here, this is Cluster W, this is the original. Then these guys here, this is an old math, and this is, there should be a reference here, but I think it's 2007 or 2008, something like this. And you have, so these are really the straightforward multiple sequence alignment, you know, the original ones. Then here, these guys here are the consistency-based aligners. So consistency, I won't have time to talk about it today, but consistency is a trick we made up, we invented at the beginning of the century to avoid some of the problems I was showing you before, to avoid some of the problem of the greediness. It's a way of making sure that all of the sequences can talk together without spending too much computation time making this possible. And that was quite striking, you know, when it came out. So Tikofi was the first one of these methods. And when it came out, it really was a clear improvement over what had been done before. And then PropCons, they re-implemented this in a Bayesian framework and so on. Then we have here something more interesting, and I will talk a little bit about this by the end of today's talk. This is something called this is consistency-based using template-based sequence alignment. This is a case where rather than aligning your sequences, you enrich them with information. And we're going to see which kind of information you can use. Okay, but whenever you see people presenting tools, there is a rule. That's number one rule. Never believe in the benchmark they do of their own methods. Never. And it's not that people are dishonest. I like to think I'm very honest as a scientist, but I'm like any scientist. I have unconscious bias, and well, anyone actually. I have unconscious bias, and it's almost impossible to be entirely fair in the assessment of your own work. And actually, one of the reasons why CASP was initiated, because CASP was ensuring double-blind, entirely objective evaluation, okay? And so, as far as all these methods are concerned, trust me when I benchmark my competitors, but don't necessarily trust me when I benchmark myself. And that's why I was so happy to see this paper coming out of Tediwan now in 2019, where they did a very exhaustive benchmark. And interestingly, they were mostly interested in evolutionary-based benchmark, which is a slightly different story. And so, but they nonetheless did something pretty exhaustive. And what you can see here is that Tcophe and Promals, which are actually Psychophe from the previous slide and Promal, they come near the top. So these are the most accurate methods currently available for relatively small data sets, because these methods are quite CPU intensive, okay? And so they did this on a couple of benchmarks, and as you can see everywhere, you have Tcophe that is not here, not on Bali-based, funny enough, because this is actually, if we had been overtraining on the data set, this will be Bali-based, but and the other ones we had never used. Actually, I've never benchmarked any. I've never used benchmark or Sisyphus, and you can see here that for some reason, Tcophe, the homology extended version of Tcophe is doing amazingly well on these data sets. Okay, so in theory, the progressive framework that I was showing you before should work like a charm on very large data sets. Why? Because, you think, and that's the image I've been giving to my students for many years. Think of the multiple alignment as you crossing a river and trying to bridge the gap between two sequences that are very distant from one another, and have the image of jumping through the river. Well, if you jump from stone to stone, you can cross a very large river. And so you'd think that if you have a lot of sequences between your two distantly related sequences, the algorithm is going to be happy. It's going to have a lot of stepping stones, and it's going to be able to produce very good models. And that's what people thought for a long time. There was even this nice result in MAFT5, in the MAFT5 paper, where they were showing that all things being equal, if you start putting intermediate sequences, you increase accuracy. So on MAFT, this is one version of MAFT, they gain 8 points. On PropCons, 4 points. On Tcophe, 6 points. On ClustalW, well, ClustalW was not so happy with this. But, on most of the state-of-the-art methods they tested at the time, it seems to work. And so what? What is going to happen? But, 100 intermediate sequences was a lot in these days, but today it's nothing. And so what if you do it bigger? And that's exactly what Desiggins did in the Clustal Omega paper. And in the Clustal Omega paper, they had a trick, which I won't explain here, to get very large reference data sets. Large reference data sets that could go up to 100,000 sequences. And then they said, okay, let's look at how accurate is the alignment with 100 sequences. Well, you get so much. And in theory, you would expect that the larger your data set, the more accurate it gets. And that's what is happening on the first few data sets. And you'd expect that these things stay flat or keeps growing up to 1, 100%. You'd expect it goes like this. You'd expect this kind of behavior. That's not what you have. You have a peak here. And then suddenly things start decreasing. Oh, that's terrible. We in-house, we reproduce this data set, and we reproduced it by, this is done, all of this thing, this is a great result, but it's a little bit comparing apples and oranges, because here you add a second data set that is different to the first one, and you average. And here you add a third data set that is different from the two ones. So yeah, there could be some artifact. There could be some data sets really dominating the trend. You think not, because it's not going all over the place. It's relatively smooth. But the right way of doing this will be to take one data set, a huge one, and to start little and to start increasing the number of sequences. We did it. I won't show the results here, and it's not so different from what you have here. So there really is a peak. The peak will depend a little bit on the family, but the fact that there is a peak is a problem, because it means that the more data you have, the less good is your modeling. It means that all this money you have spent on data is actually wasted money, because when you have too much data, it's like if you had less data. Okay, so that's opening, you know, that's the highway to hell when it comes to biology turning into a genomics-driven data science. So it's a disaster. More data equals more computation plus less accurate model. You know, it's as bad as it can get, and which means that the solution we have here turns into solution number 3. We're going to need more computers, and we're going to need better algorithms. And is there a way to avoid this? Now, we'll come to this at the end of this seminar, and I'll introduce the regressive algorithm. But before going there, I'd like to give you another bad news. You know, in biology, I've read this about, I can't remember what is the name of the guy who wrote this Scottish writer. Anyway, Irving Welsh. Yeah, there was this quote on Irving Welsh in Trainspotter. He wrote Trainspotter. They were saying in Irving Welsh novels, things are never so bad that they could not get worse. And that's definitely one thing we could say of biology, you know, and especially of computational biology. Things are never so bad that they cannot become even worse. And so big alignment, they are not only inaccurate, they're also unstable. You know, it's like this joke in this Woody Allen movie where these two old ladies, I think it's in Manhattan, where the two old ladies are talking and they're in a restaurant, and one says to the other, the food is really disgusting here. And the other one says, oh yes, it's really bad. And the portions are so small. And that's pretty much the problem we have here. These big multiple alignment, they're not only inaccurate, they're also unstable. So what does this mean, unstable? Let's say you have this bunch of sequences, you align them with method 1, and then you align them with method 2, and that will be a form of instability, you know, that you'd expect the sequences to be aligned exactly the same way, and oh, well, they're not, so what? There was a nice paper by Sushar in Science, I think it was in 2007 or 6, I should have cut it here, maybe you can see it on your screen, I cannot see it on that screen, and so he showed something very nice. It showed you take a bunch of sequences, you don't know the true phylogenetic tree, they come from yeast, but it doesn't really matter, because if you take any bunch of expected orthologs and you estimate the trees with different methods, more often than not, you're going to get different trees. That was a surprising result for a lot of people, but that's the way it is. And so it's not surprising if you know all of these multiple aligners and all the differences in their implementations. But at the time, for most of the community, and that's why it made it into Science, actually, it was a bit of a surprise. But I have worse news. It's not that it's unstable with a method. It's between method. It's also unstable between methods, within method. So if you realign the same sequences with the same method, with large data sets, you're going to get very different alignments. And what do I mean? Here is a protocol. It's a stupidly simple protocol. You take a data set, you shuffle the sequences, you align them, you do this 100 times, and you compare the alignments. And what we have here is that when the alignments are sufficiently challenging and sufficiently large, well, we have close to 50% on a guy like this, about 50% on 60,000 sequences, about 50% of the columns that are going to be differently aligned. If you think a column is a model, it means your models are unstable. Now, let me be clear. Some methods, TICOFI is one of them, they cheat so that you cannot do this. How do they cheat? They sort the sequences alphabetically or numerically or whatever. It's cheating because it does not alleviate in any way the intrinsic instability of the algorithm. The progressive algorithm is intrinsically unstable, and I will show you why and how in a minute. But, you know, in biology, whatever is unknown, whatever uncertainty is unknown to you will work against you. But once this uncertainty is known, you can domesticate it so that it works for you. And that's exactly what one should do in this case. The instability is actually a very valuable information. It's an information about the fact that some part of these models are much less defined than you'd think they were defined. You know, this portion of the model, if they're unstable, it means that you cannot fool yourself in thinking that you have estimated a correct model on these positions. And I'll show you in a minute how we harness things, but I'll show you as well that this has a significant impact on downstream analysis. You know, it's not only the multiple sequence alignment that is unstable, the underlying trees are also unstable. So how do we measure the stability of phylogenetic trees? We use something called the Robinson and Falls distance. And so think about it this way. In a tree like this, every branch is a split. This branch here, for instance, this one, it defines the subgroup JK, and it defines all of the other sequences as part of the other subgroup. So every branch split your data set in two. And you can therefore ask, when comparing these two trees, do I find the same split? And as it happens, you do find the same, you do not find the same split, you know, this K and J, no, you find the same split, but it's H. H has gone into a different place. And so one of these splits does not occur anymore, which is going to give us a Robinson and Falls distance of 2, because it is the number of splits that are not common between your two phylogenetic trees. That's how we're going to measure this. And so when we measured the tree stability, we found that the tree stability was, so 1 means very unstable and 0 will mean perfectly conserved. And sorry. 1 means perfectly stable, that the Robinson and Falls is 0, and 0 will mean very, very unstable. And what you can see here is that the instability significantly increases with the number of sequences. Well, so let me recap this. We take a bunch of sequences, we align them 1,000 times, we compare the alignments, they are different, that's what I was showing before. And then from each alignment, we estimate a phylogenetic tree, and we compare the trees. The trees are based on the same sequences, they should be identical. They are not. And the level of disagreement between the trees, as you can see here, can be highly significant. So what? What are we going to do with this? Well, what I was just saying a few minutes ago, we are going to harness this variation and to harness them into a new bootstrap value. But before I go there, it took us a while to figure this out, and it's really a cute story. Why are the multiple sequence alignments unstable? Why are progressive alignments unstable? And it's simple, actually, it's very simple to explain. So when you have a tree, you write your tree using something that is called a Newick format. It's a parenthesis. So let's parse this one together. Here we go from the root, we have a split, and so we open it. So here we have a group AB, so they're in parentheses. And then this group here is connected to another group. So this thing here, this split here, CAB, corresponds to this set of parentheses. Actually, I'd rather say this is a comma, actually. This comma here represents the split between C and AB, okay? And so every node in your tree will correspond to a set of parentheses. Now, let's look at how the aligner is going to work this out. The aligner, when it is provided with this guide tree, the aligner will align A with B, and it will then add sequence C on the top of AB, okay? Now, let's ask ourselves, excuse me, just a second. Excuse me, just a second. Oops. I need to mute myself. Just 1 second. Sorry, guys, I'm back. I had just a domestic issue. So let's go back to this. So this is our new week tree, and this is a new week tree, and the new week tree is actually a parenthesis format. And so in a new week tree, all the nodes are unordered. What does this mean? It means that this tree A, B, C is topologically the same as this tree B, A, C. So these two trees are topologically identical. The inside order of the tree depends entirely on the sequence input order, nothing else. But are they going to give the same multiple sequence alignment? Well, no, they will not. And I don't have a lot of time to explain this, but as it happens, the dynamic programming algorithm is not a symmetrical algorithm, meaning that if you align sequence A with sequence C, or sequence C with sequence A, you will not necessarily get the same alignment. Why is that? It's because in the dynamic programming algorithm, you're going to have to break ties. Whenever going in one direction is the same as going in another direction. And that will happen whenever there exist alternative pairwise alignments having the same optimal score. You only have one optimal score, but you can have more than one alignment having the optimal score. And here, you can see here that this pairwise alignment and this pairwise alignment, they have exactly the same score. But in most implementation, you will get a different alignment if you give CA or AC. So with this very simple example, what have I done? I have done something very simple. I've just shuffled the order of the sequences within the parentheses, or you can also shuffle the order of the groups, because within parentheses, you can shuffle things on the side of the comma here. And by doing this, I am actually generating significantly different alignments. One of them has 2 identities, one of them has 3 identities. So you see a lot of variation taking place here. And the reason, as I was telling you, is because of this, because of the dynamic programming algorithm that will deliver an optimal alignment, but that can deliver alternative optimal alignment depending on the order in which the sequences are provided. Okay? And so this is a reason why, actually, for all of you interested in dynamic programming, this is a formula of dynamic programming. And so here you're going to have, you navigate through this by selecting the best of these 3 values. And these 3 values are the parent cells of every cell. But if 2 of these values are identically good, you're going to have to decide arbitrarily. And that's this kind of things that make it that when you invert the order of the sequences, you invert the order in which you'd be breaking the ties. You see, if I say, so here, if I say, I'm going to, whenever the 3 possible parent cells have the same score, if I say top first, then left, then diagonal, well, if I invert the order of the sequences, the top first becomes with respect to this sequence rather than that sequence, and the alignment will effectively be different. Okay? Does this have an impact? Yes, as it turned out, with a very simple shuffling of our guide trees, a shuffling that did not change the topology of these guide trees, we were able to recapitulate about 2/3 of the tree instability that I was mentioning before, and about 2/3 of the alignment instability as well. Sorry, 2/3 of the alignment instability. But that turned out to be the same for the trees. Okay, so to sum things up, MSA accuracy do not scale up very well. Large MSAs are unstable, and trees are accordingly unstable. So what can we do? And we have to ask ourselves, why are very large progressive multiple sequence alignments so bad? The first reason, and here it's me hand waving, because these things are very hard to prove, and we were not really able to prove them in a formal way. But first of all, aligners are based on profile-to-profile alignments, and I'll show you in a minute what it implies. The second thing is that very large profiles contain mostly gaps. It's amazing. You know, if you try to scroll them, if you try to scroll one of these monster alignments onto your screen, you will just see gaps. And the screen will go on, and every once in a while you will see one residue, and then pages and pages and pages of gaps. Actually, when you store these alignments, you're actually mostly storing dashes. It's depressing. In very large columns, mutual information is lost, and I'll show you what I mean here. Let us say that this is a bunch of related sequences. Okay, not the most exciting sequences, but they are, let's say they are related. Okay, and we found X and Y, and in other sequences we found Y and X, Y and X. You know, this can happen. This kind of pattern you may find if you have a salt bridge. You know, a glutamic acid with an arginine, for instance. So they have minus plus, and so they will not induce a fall, but they can be convenient to stabilize the inside of your protein. Okay, and if you look at enough instances, you will see from time to time that the salt bridges are actually inverting. You will have the arginine turning into a glutamate and the glutamate turning into an arginine. Okay, but the profile doesn't know about this. The profile is simply going to average this thing. So the profile will be very happy to find proteins that look like the ones you had before, but it will also be very happy to say that these will be so it will be very happy to find these guys, but it will be just as happy and give a score as good to things that are putting Y and Y together or X and X together, even though you've never seen these proteins, and it's quite unlikely that these proteins exist or are members of the family. And I call this effect flattening, because in effect, the profile is flattening the information contained in your sequences. One of my impression with something like AlphaFold 2 is that AlphaFold 2 is actually able not to flatten this profile information. And that's one of the things we're actually working on these days, trying to demonstrate this. And I'll show a few examples later. Now, the bottom line is that profiles do not care about covariation. You know, covariation becomes invisible in profiles. And that's a shame, because covariation is one of the strongest signal we have access to. You know, covariation, there are many mysteries that biology try to solve. And most people my generation try to solve the protein folding problem. As it turns out, on proteins, this was not so easy. But on RNA, well, we have one of the major victories of modern biology, because just doing alignment, people like Robin Goodell were able to predict very accurately the secondary structure and some of the tertiary structure of ribosomes. And that was done by just using covariation, just doing alignments and looking at covariation, looking at positions with a lot of mutual information. So it works, and we lose it in profiles. And that's, in my opinion, one of the reasons why the profiles actually, the very large progressive alignment fail. Another aspect is that ambiguous tie breaking can really break the DNA, the multiple sequence alignment. And I'll show you an example here. Let's say that these are sequences that are otherwise different, and that's why they cluster apart. And we can tell that we'd like these two subgroups to be aligned the same way. We'd like either this A to be to the left here, or we'd like this A here to be to the right, which will reflect conservation and make it much easier to classify our sequences. But depending, and because of the arbitrary nature of the way ties are broken in the dynamic programming, we have no way to see, to control this. And when we reach this merging here, we may be in a situation where our residues are all over the place. And actually, this is precisely the effect that occurred with when we were working with Fyodor, and the reason why even closely related sequences were very difficult to align correctly. And as it turned out, if you have enough sequences, these kind of patterns here are going to have a terrible effect and are going to make you think that you have duplication. They will generate spurious duplication, what looks like duplication, so spurious duplication. Oops. Okay, so I had known of these things for a very long time because earlier in my career, when I was working in Marseille, we were trying to make very large multiple sequence alignment of kinases, 500 kinases, a human kinome. And you know, on this case, you know very well your active site. You know where is the active site. You know where is the ATP binding pocket. You know, all of these things are pretty well established for all the kinases. And it was almost always impossible to get all of these positions correct. There was always one going to the left or going to the right. And the irony is that whenever you were randomly selecting 20, 30 kinases, the alignment was always entirely correct. So this really was something that had to do with a size effect, with the piling up of all of these sequences. There was only one tiny glitch that was causing your model to fail, which is really the inspiration behind the algorithm I'm now going to introduce, which is the regressive algorithm. And so what fixes did we incorporate? So the gaps. We have to avoid aligning very large data sets because the very large data sets, they are drawn in gaps. The sequences are drawn in gaps and there is nothing to be seen. All the information becomes extremely difficult to manage. The flattening, what I was talking before, the fact that profiles do not see covariation. And then the ties, these early decisions that are done on closely related sequences, they can never, ever be, you know, when you try to synchronize, think about your progressive alignment growing from the bottom to the root. You know, you start with very closely related sequences, and there is no way you're going to figure out a way to synchronize information across your entire data set. And that is kind of a killer here. And so what? So we implemented something called the regressive algorithm. How does it work? It starts with a guide tree, like any algorithm, any sequence algorithm I can think of. So here, I played a trick. I named every sequence by its length. So it's just for the illustration. So the sequence that is named 2 is actually 2 residue long. The sequence that is named 5 is actually 5 residue long, and all these kind of things, okay? Why? Because what we are trying to do here is that we're going to try to assemble a data set of size n that will be as representative as possible of the entire data set. And we are going to assume that given 2 sequences, the most representative is the longest. It's not far-fetched, actually it works quite well. There are smarter ways of estimating the amount of information contained in the sequence, but length, as it turns out, is a reasonable proxy in most cases, okay? And so here, 2 and 1, we are going to keep sequence 2, and we are going to label the parent node with the name of that sequence. Here, 6, 4, 6, here, 5, 3, 5, 11, 8, 11, 13, 9, 13, 7, 12, 12, and on and on. So we are labeling every node with the name of the sequence, of the child sequence that is the most informative. Fine. How about this node? Well, we can play the same game. Oops. We can label this node with the name of the most informative sequence of these two sequences. The most informative is sequence 6 because it's the longest. Here is 11, 13 for the same reason. And here we have to go one node before, which we'll do now, okay? And we're going to do this on and on with the longest sequence making its way all the way near the top. Now, by this measure, you realize that 14 is the most informative sequence. If I wanted the two most informative sequences, I would look at the children of the root, and I will say that 11 and 14 put together, you know, if I were to pick up only two sequences, well, if I were putting 11 and 14 by my criteria, these will be the most informative sequences. If I had used a different criteria, I'd be pushing the sequences all the way up by this other criteria, and I will have a subset of sequences that are the most informative by this criteria. Okay, great. So what do we do next? Next, we can go the other way around and we can start collecting sets of sequences, which are special because they are the most informative sets of sequences. And this is exactly what the algorithm is going to do. So let's say that we want all of our multiple alignment to contain the three most diverse sequences. Well, I am going to unfold the root. It gives me two sequences. That's not enough. I'm going to do this generation-wise. So once I'm finished unfolding this layer, I'm unfolding the first node here, 6 and 11. So that's 1, 2, 3. Now at n equals 3, I stop. I have collected one data set. Okay, it's here. It's going to be the parent alignment. What is great with this alignment is that, yes, it's going to be a bit challenging because the sequences are the most distantly related. But the decisions taken here are going to be imposed on all the children, which means that by doing this early alignment, I'm actually synchronizing all the children. And that's what matters here, this synchronization. So the problem of the ties that may not be synchronized between your left and your right tree, the problem of the ties is solved, okay? And now I'm going to go one generation below. I'm going to go one generation below, and here I'm going to generate here another bunch of another alignment by simply opening 6. And so I open 6, it gives me 2 and 6, only two sequences. I have to open a little bit more. I'm going to open 6, I'm going to open 2, 1, 2, 3. That's done. I don't have to open anymore. And that allows me to generate the first generation children. And I'm going to generate the last generation children. But here, most of the cases, I have only two sequences and they are terminal. I cannot open anymore, okay? And so these are my second generation. What is cool is that all of these multiple sequence alignment can be estimated independently from one another. I don't have, they can all be computed at the same time on a big computer, and you send everything to different nodes, okay? And of course, in terms of parallelization, that's a dream, okay? So, but once you have all your alignments, once you have estimated all your multiple sequence alignments, you still need to do something. You still need to merge your alignment. And here we have a trick, because as you go down from the parent to the first generation to the third generation, you can see that you are maintaining a common sequence, a sequence that made it from the root, from the tips all the way up to the roots. And you can use this common sequence to drive the merging. It means you don't have to align profile to profile. You're just threading your parent sub-alignment with your children's sub-alignment through the common sequence. And in fact, what is cool is that you can do this in a very, very efficient way by simply matching the indexes and keeping the length of the gaps in memory, meaning you don't have to write a big alignment. You can only write the correspondence between the columns. And that saves you a huge amount of disk. Think about it this way. If you are aligning some of the data sets we have here, the largest data set we have here is 1.5 million sequences, which is about, on disk, every sequence is about 200 amino acid long. So that's about, about 300 megs, okay? If you align it it contains so many gaps that it's going to take 4 terabytes of data. And these 4 terabytes, the problem is that I was saying on your disk, that's not on your disk, that's on your memory. That's a memory footprint. Because if you store the gaps, if you do it the regular way, you have to put the gaps in memory. But because of the way we are computing our alignments, we don't have to do this here. We can simply count the length of the gap, and you can keep every gap as an index. And this saves huge amounts of memory. And as those of you who are computer scientists in this audience know, if you save memory, you save time, because it takes a lot of time to allocate memory. And when you start running out of memory, it takes a huge amount of time to allocate even tiny amounts of memory. Okay, so third-party tree, the cool thing about this is that we can use any tree we want. We can use any tree estimation method we want, the cluster omega trees, the math trees. We can use any aligner we want to do these little guys here. You know, we don't have to use T-Coffee. We can use anything. And this is very fast because overall, the merging is linear with sequence lengths. And because the gaps are not stored in memory, it's even faster. Now, does it work? Well, how do you measure the accuracy of very large alignments? Now, this is something that was pioneered by Desiggins. This is called embedding. Because you don't have a structure, or we use not to have a structure for every sequence, what you're going to do here is that rather than having a reference data set with a very large number of structures, you throw in a collection of homologous sequences, all of the sequences you have having a structure that are homologous. So for instance, you have PFAM, you have a PFAM domain for ABC transporter, and you're going to throw in the 20 structures you have for ABC transporter. For these guys, you have a reference alignment. And so when your alignment is done, when you have finished aligning with the regressive algorithm or anything, you extract your structures, the projection of your structure, and you ask, how were the structures aligned within the large alignment with respect to the reference alignment? Okay, and you know, it's exactly what we were discussing before. If 5 and 7 are nicely aligned, then you're happy. And if 9 and 8 are differently aligned, then you're unhappy, and that gives you a score. Okay, so here is the result. Let's read this table together. This is a situation where I have used as a guide tree Partry. Partry is a very fast tree by MAFT, and I have used as an algorithm a fast Fourier transform, number of NS, I don't know, the S stands for 1, so number of iteration. So FFTNS1 is one of the most accurate aligner we have. Okay, and so if you do this, you get an accuracy of 29.64%. If you use MAFT as an aligner and the same tree, but in the regressive setup, you reach 35% accuracy. Embed and MAFT, you lose a little bit in accuracy. Partry and Clustal O, you dramatically increase the accuracy. Embed and Clustal O, you significantly increase the accuracy. Okay, and overall, by applying the regressive algorithm on your data set, more often than not, you are going to increase the accuracy. Okay. Now, interestingly, if you were directly aligning the sequence of the reference, you will almost all the time get a better alignment. And that is something that is quite sobering. It means that in reality here, because we have all of the sequences, we should have 51% accuracy. But in reality, having extra sequences means that we have not yet managed to bridge the gap, the issue of the progressive algorithm. We're improving significantly over this issue, but we are not yet solving the problem. Yet what is quite nice here is that we can now deploy onto these data sets aligners that will be unable to deal with data sets of that size. And that is especially true of GINSI. GINSI is the most accurate version of MAFT. It's a version of MAFT that uses a little bit of consistency to do better alignment. SPARE-SCORE is the latest version of MAFT. It's by far the most accurate. And as you can see here, when we apply, so on GINSI, I don't know how accurate it is in non-regressive because you cannot do it. But what I know is that once we deploy GINC with part3 and embed, we become able to generate significantly more accurate alignments than we were with the other aligners, okay? And here we are getting so here we were 10 points away from the reference, and now we are getting quite close to the reference, meaning that even though our structures are embedded in very large alignments, we are nonetheless managing to align the structures, the embedded structures, as accurately as if we were aligning the structures of their own, okay? And so that's very encouraging. Sometimes it runs faster, sometimes it runs a little bit slower, but overall it's pretty good. These are pretty good performances. And of course, this is much longer, but bear in mind that here you will need months, years of computation to run part3 on data sets of that size. So that's a clear improvement. We have here a graph where it's nice because since you're using the same aligner and the same guide tree, you can ask, you can represent the evolution of these things. And most of the time we run slower, so that's CPU time, and we are more accurate, okay? With a few exceptions here, where on embed we run a little bit slower, but we are quite significantly more accurate. And on this one, on FFT and S1 embed, we were a little bit less accurate. That's the only issue where we had a drop of accuracy. Now, does it scale? So on the paper, we were able to show that we can, so here we don't have the accuracy because we didn't have the reference alignments here, but we were able to align the largest data sets available at that time, about 1.5 million sequences, which no other alternative method was able to touch. Here you can see the non-regressive ones at the time. And the cool thing is that it really scales very nicely. You know, computationally speaking, you could align as many sequences as you want, and it's going to be roughly linear in time. So that means you take an algorithm like MAF-GINC that has a very complex, a very bad complexity, and you immediately turn it into a linear algorithm. Okay, that's just another representation of this thing here. Okay, so we published this thing in Nature Biotech a few years ago now. It's all available for free, and we'll be happy to help anyone who wants its open code and everything. You know, it has all the hallmarks of these things, and we'll be happy to help anyone who wants to use it. So what next? So as we saw before, the real issue is accuracy. We'd like to be very accurate. And the highest accuracy we could get was with these template-based sequence alignments. And so will it make sense to bring the regressive algorithm into a template-based framework? And the answer is obviously yes. Let me explain a little bit how this template-based thing works. You know, let's say we are trying to align these two sequences here. And leucine is one of the less informative residue you can think of. This could go this way or this could go that way. So, but if we run a BLAST or the kind of multiple sequence alignments that AlphaFold 2 is using, we're going to see that these two leucine here are different. This leucine here is very highly conserved, which is typically what happens with the leucine zipper. While this leucine here is your average leucine. It can turn into a valine, an isoleucine, and it doesn't mind. And so without this information, you will not know which leucine has to be matched with leucine. But with this information, you know that there is only one correct alignment, and it is this alignment. This thing here, this profile 1, is a template, and the profile 2 is a template. And how do they work? They work with TICOPHY. In TICOPHY, we start by doing all the pairwise alignments, and we turn this in what we call a library. And the strength of TICOPHY is that you can have as many templates as you want. Here is Expresso. Expresso is the mode of TICOPHY. It's very popular to make multiple structural alignments where you give your sequences, and rather than aligning your sequences, you look for a template with BLAST. You align your template with your favorite structural aligner. We use BLAST, we use SAP, you could use any, and then you get a structure-based alignment of the templates. Since the templates are aligned with the sequences, you just remove the templates and you have something that is a structure-based alignment of your sequences, okay? It's very powerful, it's very simple, and it's based on this template-based paradigm where sequences are replaced with templates. Now, does it work? Yes, there was this nice benchmark, not by us, by Mathilde Carpentier a while ago, where she shows that this is, so these are the sequence aligner. This is RMSD, so this is a measure of structural accuracy. And so in red, you have the sequence-based aligner, and in blue, you have the structure-based aligners. And TICO-Phi is somewhere in red. No, yeah, TICO-Phi is, sorry, the other way around. In blue, you have the sequence. No. I'm losing it here. Yeah. So you have TICO-Phi, and then 3D TICO-Phi should be here. TICO-Phi SAP. They called it TICO-Phi SAP, and you can see that TICO-Phi SAP is somewhere mixed within the blue things you have here. So, and it has behavior. It has mostly the behavior of a structure-based aligner. Okay, so now things have changed a lot, and that's the reason why I named this talk post-sequence, multiple sequence alignment in the post-sequence era. We can have all the structures we want now. AlphaFold is amazingly good on almost everything. It's not super great on transmembrane proteins yet, but on most, almost everything that is globular and non-disorder, it's amazing. So can you base your multiple sequence alignment on this? And this is just a preliminary result. We are writing the paper right now. And what we were able to show is that if you take a bunch of sequences and you align them, you're going to get a sum of pairs score of about 70%. If you run Psycho-Phi, you're going to get a score of around 77%. That's what we knew. And if you take AlphaFold 2 model, you're going to get something that is going to be 93% similar to what you'd be getting if you were basing your alignment on the bona fide PDB structures. That's an amazing result, right? These are just sequences. These are the most accurate sequence alignments we were ever able to produce. Now the trick here, it's not an easy figure because, as you know, AlphaFold is trained on PDBs. And so in order to do this kind of benchmarks, you have to carefully select the PDBs on which you'll be working so that there is no circularity. And that's actually most of the work in this project got into this. Okay. Now about trees. So as I said before, if you cannot beat them, you have to join them. So can we use the instability to make it work for us? And that's Unistrap. And I'll be finishing on this. And so the idea of Unistrap, which is based on what I was showing earlier on tree instability, on the bad news of tree instability. The good news here is that you can use this instability to increase the informativeness of your bootstrap value. Do you guys remember what is a bootstrap? I have a multiple sequence alignment and I can estimate a tree from this multiple sequence alignment. And then once I have estimated the tree, I have a tree, but I don't know if the branches are very well supported by my alignment. And so Felsenstein, 40 years ago now, 30 years ago at least, worked out, I think it's in 80 something, worked out, that's 40 years ago, yes. He worked out a great protocol where you estimate your tree and then you're going to resample your columns. You draw, put back until you regenerated an alignment the same length as the original alignment. You estimate a tree and you do this about 100 times or more. And then you count how many times each branch has occurred in your 100 replicates. These things are called replicates. Okay. And if a branch occurs in all the replicates, usually it's quite trustworthy. If a branch occurs only on a small fraction of the replicates, well, you may have a problem. The cool thing here is that it's very easy to plug in our tree, our alignment instability in this protocol, rather than drawing columns from the trees, from the MSA. You can re-estimate your MSAs. And so you can combine the MSA re-estimation and the tree re-estimation. And so you can have replicates that simultaneously take into account the MSA instability and the tree instability. And that's what we did in Unistrap. And we have something quite cool on this very large data set. You can see here that most of the time, the bootstrap branch support is superior to the Unistrap branch support, which means that effectively, we convincingly show here that the bootstrap, the Felsenstein bootstrap, is overestimating the branch support. And here we effectively decrease this overestimation. And on some of these things, it's quite significant. You know, you have branches, you have a lot of dots in this corner. So these are corners where you have a very good Feldenstein bootstrap support and a very low Unistrap bootstrap support, meaning that you think you can trust your branch, but you cannot. This branch is not trustworthy. It's not trustworthy because it is based on a part of the alignment that is obviously unstable. Okay, so is it worth using it? This is just what I was saying here, you know, false positive and false negative. The branches that you think are correct in Felsenstein bootstrap turn out not to be correct. Now, this notion of correctness in big data is more important than ever, because in big data, you will always find sufficient data to support any hypothesis. Okay, and so you cannot fool yourself with this, oh, this is supported. No, you really have to figure out how stable is the support you have for your hypothesis. Now, I'll finish with a small parenthesis on our most successful output of this last year, which is Nextflow. Many of you have probably already heard of Nextflow and are using it. This is something that has been surprisingly successful in the lab. It's a pipeline language, and it's a nice story because it really is something we wrote for ourselves. It's not something we wrote for the entire community, but because we are a small lab, we had to write something very simple that will be usable by every member of the lab very rapidly. And this guy in my lab, Paolo Di Tommaso, did something amazing. He did this on his own. He wrote this language. He implemented everything on his own. It became amazingly successful in the lab, which meant that my students were using it. And this guy, Evan Floden, who turned out to be, who was merely a PhD student in the lab, working on a different thing, he just jumped onto Nextflow and he figured out that Nextflow was allowing reproducibility. And suddenly, from a mere pipeline language, we had something that was actually increasing stability, increasing reproducibility in biology. Evan was one of the PhD students Rita gave me from the Bologna master. And so this became the beginning of a very nice story, and they have started. So actually on this paper, I think I'm the only one who didn't start my own company. So Paolo and Maria, Paolo and Evan started Sequera Labs, that has just secured three Chan Zuckerberg Initiative grants now. So that's something very, very promising. And Maria Chatsu and Paolo Prieto went to the UK and started their own company, LifeBits. And I understand that they just secured a huge grant in their second seed of rounding of 40 million euros. So these guys are doing amazingly well. And I like to think that this was all the result of the hard work we did together in the lab on Nextflow. And more recently, the latest development on Nextflow is the NF Core framework. It's like a standard for Nextflow that is making the pipelines highly interoperable. And this was really driven by Phil Ewell from SciLife Lab, and of course Paolo Di Tommaso from my lab. And that has been a very successful development of Nextflow. Okay. And so let me conclude, because I've been talking nonstop and many of you are probably asleep in front of their screen now. I apologize. So the regressive algorithm, it's faster, it's more accurate, and it's more versatile. And in not too distant a future, thanks to the template-based paradigm that I was showing you before, we hope to be able to integrate all this within the realm of the AlphaFold 2 predictions. And so that's something that is going to have a huge impact on sequence alignment, because now we won't be using sequences anymore. We'll be using structures to align the sequences, predicted structures. This is really the way to go. Unistrap, instability. Instability is everywhere in biology. It's everywhere in data science, in bioinformatics. You have to identify it and you have to use it for your own benefit. Instability is essential to figure out what is trustworthy and what is not trustworthy in your modeling. And with huge models we are not building, this is more important than ever. A big thank to all the collaborators and all the people who have been working this. Lots of people are missing here. This will require a much longer slide. But thank you all for your attention, and sorry for being a bit long.
Thank you. Thank you very much for the beautiful lecture, the very comprehensive lecture. So I ask if there are questions from the audience. I invite the students of the master to ask questions. By now, Rita.
As a student, Rita, you're a second or a first year student?
Yes, I'm a student in this field. Oh, fantastic. No, by the way, if somebody else wants to ask questions, I can be the last one. No problem. But since I don't see so many hands, I go ahead. Okay, so first of all, as to AlphaFold, in my experience, just in teaching, because I don't do anything with AlphaFolds myself, AlphaFold is very good if sequences and structures are already fairly well known in the PDB databases and eventually also as information overall, etc. When you are dealing with very long sequences and practically when you don't know so much about the protein family of these sequences and more, first of all, you don't have the structure. Even AlphaFold is, fairly suggesting that they are sort of not so well in the prediction, because you have regions in which you clearly see that their own scoring system is somehow very low. Second of all, AlphaFold, when you are dealing with multi-domain proteins, particularly at the level of domains contacts, AlphaFold is not working perfectly well. For sure, through the years, they will improve and they will give us the possibility of, predicting whatsoever. But at the moment, there are situations in which it is a problem to use AlphaFold predicted structure because you don't have any experimental counterpart to even to suggest whether they are right or wrong. So my question is, are you considering, to use AlphaFold with specific, for example, length of the proteins? Do you have a sort of, framework in which AlphaFold is for sure considering, it is considered a sort of gold standard predictors, or are you using AlphaFold with any length, or do you think?
Yeah, no, okay. This is, this is almost the Olympic Games. These are very, very precise exercises. And so we work on domains because this is where we have the reference and because we have the proteins and all these kind of things. But it's not a big problem because at the end of the day, you don't want to align multi-domain proteins in a Go. You want to chop them into domains and you want to work domain by domain. But I have something to say about AlphaFold. And I think my expectation is that there is more than meets the eye in AlphaFold. And what I'm saying here is that I don't think AlphaFold is a structural predictor. I think AlphaFold is more than that. I think AlphaFold is it's a super profile. These are profiles that will not flatten information. And once it has processed the multiple alignment information, it generates a model. And because this super profile is so good, more often than not, the structural model is good. But even when the structural model is not good, it reflects something of the biology of the proteins. And I think we can do very useful things, even with the AlphaFold models that are actually not good. And that's one of the things I'll show you and we'll discuss probably in Bologna in September, I hope, or when you come here in Barcelona, because there is something quite interesting behind this.
No, I may agree with you. Problem is that sometimes, you're opening up UniProt files and all of a sudden you see all these drawings and they don't make any sense at all. But this is obviously if you're going into details and see what's going on with AlphaFold.
Sorry, Rita, if you're talking about, does it make sense to pretend we have the structures of everything and to put them in database
fantastic.
like they were real structures.
Okay.
And if you think it's a crazy idea,
So
the
idea is that crystallographers or other people working with NMR or with whatsoever methods to determine structure, they have a lot to go and a lot to do in any case. Okay, now, and second question, perhaps more interesting, is the following. To which extent scoring matrices are there, and because, you know, we have these spectacular scoring matrices since, I would say, 30 years, and they are still good, and they are implemented in any tool that we are using routinely. How do you feel about them? Do you use them? And to which extent you do prefer one scoring matrix to another one, etc.?
Well, you know, it's a funny thing, scoring matrices. So it's a mathematical object, right? And so it got mathematicians very excited in the early days. And you have hundreds of scoring schemes, hundreds of alternatives to Blossom. And I think I've often heard said that the Gonnet was the best one, but they never really figured out the tuning of the gap penalties, and so people didn't use it much. I tend to use Blossom 62 like everybody. Yeah, but if you ask me, one of the things I expect to come out of deep learning are actually much better substitution matrices. Because Facebook has done this a couple of years ago. They learned all the proteins in UniRef, and if you learn all the proteins in UniRef, in theory, you could have substitution matrices. You could work out models that take into account epistatic relationships, and that's one of the things we want to look at. And my expectation is that in not too distant future, we'll be using deep learning based substitution matrices. Well, you know, but one of the things, and I don't know, I mean, you started on this deep learning much before everybody here. And I'm still confused about whether deep learning is actually learning things by heart or generalizing, and whether it is generalizing or learning by heart, and whether it's possible to draw a line between these two things. And, you know, think about AlphaFold 2. Let's say maybe we don't have so many protein fold in the end, and maybe if you learn everything by heart, well, you can tell about the neighborhood, and we maybe miss still a few hundred structures, and when we will have them, and it's okay, we don't understand anything because we have learned everything that was to be learned by heart. But it's not quite the same as having learned the principles, right? And
yeah, true.
It's very difficult to draw a line and to know if deep learning is actually generalizing or learning.
Well, I agree with you. There are people who think that deep learning is to learn by memory, and they are using it in this way, and obviously then generalization is over. And the line that separates learning by heart or generalizing through learning is a little bit difficult to achieve, particularly when, you know, but there are several papers around and communities trying to set up rules in order to, well, that needs to be followed in order to produce paper in this field. So I highly recommend always to follow the rules, because JMB and other papers are also having a sort of policy when you are using deep learning or machine learning in order to produce data, and you have to comply with them, otherwise the journal will not accept your data. So this is really difficult. But even though you have, I don't know, because I'm not familiar with these matrices generated by deep learning, so I really don't know how they work and if they are reliable or not. Perhaps you know.
They don't exist now.
Okay, still they don't exist. Fantastic.
But I think this is something that could really be worked out.
The dogs. I can hear the dogs.
Yes.
Okay, so we will have plenty of time to discuss this interesting aspect of deep learning versus multiple sequence alignment. Hello?
More questions from the audience?
Where are my students? The students of the master.
And also the PhD students.
Okay, fantastic. But, you know, since I don't know many of the people.
I have a question that maybe you know. AlphaFold is based on multiple sequence alignments. How are these alignments built? You mentioned that probably they avoided the flattening problem, but I didn't understand how.
So that I don't know, and we have to look deeper into this. But they are not the kind of multiple sequence alignment I was showing. These are really very fast database search, and so this is what this guy Martin Steininger did, and this is just super fast scanning of the database to bring all the homologous and everything. And how the flattening is avoided, that I don't know, and it's buried in the details of the neural network. But somehow the neural network figures out a way to use these alignments so as to avoid, because if AlphaFold was losing the epistatic relationship, it could not, there's no way it could work. And so by definition, and in fact, a neural network really has a capacity to tease all of this information and to maintain all this information. It has a capacity to keep this in memory. And I think that's what they do. But we are doing experiments around AlphaFold exactly to figure this out and to try to look at these things.
Okay, thank you. More questions?
Thank you very much for this lecture.
Well,
so you are coming.
Mamma
mia, what happened? Mi sono disconnessa.
No.
I'm still connected?
Yes, you are.
Okay, so I don't see anybody else, but I wanted to remark the fact that in September you will be teaching with us, to us.
I hope so.
Yes.
I hope so. And I hope it will be presential. It will be great.
Yes, we hope.
And, we hope so.
And so if anybody has any question they want to ask or anything, do not hesitate to drop a mail, whether a question on the next flow or a question on the regressive alignment or anything. Just feel free to drop a mail on anything that would be interesting. And aside from this, thank you for your attention and for being a nice Zoom audience. And I hope to see some of you in September.
And congratulations for your work. Okay.
Thank you very much. Thank you.
Bye-bye.
Bye-bye. I remind everybody that the next lecture will be on Tuesday afternoon. So Tuesday afternoon at 3, Professor Jones will give the next lecture of the school. Thank you, everybody. Bye-bye.
Bye. Thank you. Bye.